SCA working group results. So as Thomas mentioned, there have been those working groups getting together and looking into the what actual work we can do as a SCA Global to facilitate the clinical trial readiness in an international setting. So Thomas, please go ahead.
Okay, it's my pleasure to start with the presentation of working group results. And the first working group that I am leading is the one on the clinical outcome assessments. And before presenting the results, I would like to share a few considerations of the urgent need for harmonization and standardization. So I will start with that. Why do we need consensus on outcome measures, whether clinical, MRI, or biochemical? First, as we mentioned in the beginning, our long-term goal is to really have longitudinal projects endorsed by SCA Global or ARCA Global. And one possible example that we were dreaming of from the beginning is a worldwide registry according to the role model of the Enroll HD. And I think it's very obvious that you can start such a initiative only if you have a consensus on outcome measures. What we are currently doing, and this is already getting quite fruitful, is sharing data. And you are only able to share data and do meaningful analysis in different cohorts if you are using the same outcome measures. And we are in the fortunate situation that for a number of large cohorts, the ESME, the CRC, SCA, EuroSCA, and RISCA, there was a core assessment including SARA, which was the same in all cohorts. And this allows then these merging of cohorts and doing overarching analysis, as was presented by Michael Lübachs in the flash talk in the last session. Most important reason to agree on outcome measures is the preparation of trials. And here, what we're currently doing in this meeting is to reach consensus among academic investigators and also industry, but this is only the first step. The next step, and maybe the more important one, is then to reach a consensus with regulators. It is not very useful that we all agree that SARA or INAS is a useful outcome markers, but then the FDA or EMA says they don't accept it for trial. So now in this meeting and in the next meeting, we have to reach the consensus in this consortium, but then this is the basis for discussing that with regulators, and we hope that the critical path initiative will be helpful for reaching this final consensus that we need for the trials then. Now, how does the proposal look like for the clinical outcome measures for SCA Global? We decided to have a structure consisting of a minimal data set, and the minimal data set is a data set that can be obtained during a routine clinical consultation. So this does not require any study visit or additional funding. This is so focused that you can obtain these data during a routine clinical consultation without a lot of additional time effort. But then we are additionally proposing an extended data set, which is understandably more demanding. It's typically collected in a study visit, takes more time, and this extended data set then is particularly useful for research purposes, although this minimal data set, I believe, is already very useful if you if it's applied and you come up with really large numbers of patients and also longitudinal data. Now, how does the minimal data set look like? I think one very obvious thing is you need to identify the person, the patient. You need an identifier. There should also be some information about participation in previous studies and registries. So, for example, if we include patients who previously have been studied in EuroSCA, we should know this, so the study acronym and the old identifier should be documented. We need some core demographic data, sex, age, and these things, and we must very clearly define the genetic category. So in a participant with a known SCA genotype, for example, SCA3, it must be stated whether it's a mutation carrier or if it's a first-degree relative with a 50% risk, which has not yet been tested, or if we have someone from a SCA family with an unknown genotype, there should be the information about the negatively tested genes and the information whether this person is ataxic or a first-degree relative. And it's always very useful if we think of future registries to include unrelated controls. Here it would be useful to know whether it's a spouse control, which is often used in these studies and registry, or a community control. Then we need the information about the genotype for the polyQ or repeat scars, the repeat length of the normal and expanded allele. And for those with conventional mutations, we need information about the type of mutation. Then the minimal data set should include the disability status, and this is the disability staging that Jeremy Schmarman also mentioned in his talk today. The 5 stages, starting with a normal, then ataxia, but independent, permanently dependent on walking aids, permanently dependent on wheelchair, and death. This is extremely useful information, highly relevant for the patient and easy to obtain. We suggest also to include patients' global impression, because this is very helpful for estimating the clinical relevance of scale changes. And this is a very simple question. You ask the patient whether the disease status is worse or stable, or in some cases even improved within the last year. And there was consensus in the working group that the minimal data set should include this scale for the assessment and rating of ataxia. There are a number of other rating scales around, like ICARS, VARS, and NESCA for SCA3, but there was little discussion that the SARA is the scale which has the widest distribution and where we have the largest treasure of data. So it was easy within the working group to reach the consensus that in the present situation we should have the SARA in the minimal data set. Now I move on to the extended data set. Here we suggest to include comorbidities and medication as an additional measure of ataxia severity, a timed test, the composite cerebellar functional severity score. There is an alternative, the SCA-FI, but the opinion of the working group was that the composite cerebellar functional severity score is easier to handle. It's just two tests which can be performed by the patient. For the SCA you need three subtests, the walking, the 9-hole pegboard, and the speech test. And the CCFS results are age controlled, and in SCA it's always quite complicated calculation of Z-scores, which make it a bit more difficult to handle. We also believe that it was necessary to include the INAS, the inventory of the non-ataxia signs. We already heard something about the INAS today. The INAS from the beginning was not planned as an outcome measure, and consequently it is not really sensitive for change, but it's a very useful documentation which non-ataxia symptoms are present or not present. There was also consensus that it's a measure, a patient-reported outcome is absolutely necessary. And what was available or is currently available is the ADL scale that we used in the EuroSCA that we simply took from the Huntington's disease rating scale, and alternatively the ADL scale that comes from the Friedreich's ataxia rating scale. And these both ADL scales are very similar, so there are no big difference, and the suggestion and consensus within the working group was to use the Friedreich's ataxia rating scale. And in Europe we also changed to the ADL scale from the Friedreich scale in the ASME, in the ongoing ASME study. We're using the FARS ADL. Depression is important. In many SCAs we have very new data from the ASME cohort where we see that 2/3 of the patients have depression. We also know that it's progressive over time, and here the patient health questionnaire PH9 is very useful. And as a measure of cognitive and affective involvement, we suggest to use the Cerebellar Cognitive Affective Syndrome Scale developed by Jeremy Schmarman and his group. One additional comment with respect to the ADL scale, we heard at this meeting about the new PROM ataxia by Jeremy Schmarman, and this is a very carefully developed new ataxia specific patient-reported outcome measure, and it may well be that in the future we will replace the FARS ADL by this new PROM ataxia, again developed by Jeremy Schmarman and his group. So this is in short our assessment program, clinical assessment program that the working group is suggesting. And with my final slides, I would discuss a bit more in detail the SARA score. As you learned, as you know, and again learned from my presentation, the SARA is a kind of core assessment in the whole field. So we have to bear, it's very important, and we have to be very critical about this clinical scale. Most of you know SARA, so it's a clinical scale that is based on semi-quantitative assessment of cerebellar ataxia on an impairment level. It has 8 items. It's short, you can do it in 10-15 minutes in patients, and it's quite easy to apply. It has been carefully validated. A number of publications are available on the validation of SARA, and this is the scale where we have the largest amount, both of cross-sectional data, but also longitudinal data from Euroscar, other natural history studies like SCA, CRC, SCA, and in principle, there are also data, although they are currently not available, from placebo groups from drug trials. Another advantage of SARA is that it has been translated into various languages. It may be that these translations are not very important for research purposes, because in the research setting you can use the English version, but SARA is now in worldwide use in patient care, in routine patient care, mainly applied by physiotherapists. And for these application, it's very important to have the SARA in the local native language. And there's a very useful web site given here, where you have a review of the whole SARA scale, and you have also bibliographic references of available translations. And again, what is more important for research purposes, we have, and this is mainly work of Markus Grober-Einzler, and this was done in a collaboration with Tübingen, Matisse-Novzig. We have developed an online SARA training tool with a certification option that is free to use for all SCA and ARCA Global members. So a lot of reasons to use SARA. On the other hand, you know that SARA is not undisputed. There is considerable critique concerning usefulness and relevance of SARA, and I'm just listing here a number of arguments questioning the usefulness and relevance of SARA, and these arguments were mainly raised by regulators when protocols for drug trials were submitted to regulators. There is strong intra-individual variability. There's the example of the Biohaven study, where there were large differences between the screening visit and the baseline visit, which was only a few weeks after the screening visit. This is something that needs to be discussed, but we will present data in this meeting. This is in the next Flash Talk session presentation by Markus Grober-Einzler, where we have recorded SARA at home. And what we see there, that in all patients, there is a considerable variation from day to day in SARA. So there is some degree of natural fluctuation of ataxia severity, and this appears to be a feature of the ataxia and not a problem of the SARA. There's questionable functional relevance of single items, specifically of items 5 to 8, hypermetria, dysdiadochokinesis. So these are all neurological constructs, and the SARA was devised more than 15 years ago, to say it very simply, by neurologists for neurologists. We wanted to derive something from a conventional neurological examination, and we did not consider seriously enough whether all items and changes in the items are really relevant for patients' everyday life. And this particular applies to the items of the appendicular ataxia, so the hypermetria, the fast alternating movements, and the tremor. We know from the natural history studies that there is variable sensitivity to change of single items, but we saw the data from Friedrich's study from Massimo Pandolfo. So this may depend on the disease. For example, in Friedrich's ataxia, you have a ceiling effect for the gait and stands, but then in later disease stages, there is higher sensitivity to change for the items reflecting appendicular ataxia. We have different scoring ranges, which has been criticized. Items 1 and 2 require more than one distinct task. And in particular, item 7, the fast alternating movements are particularly difficult to rate because we have more than one criterion for one response category. So there are a number of points which have to be considered. And my personal opinion is that we are now in the situation where we have to seriously consider a modification of SARA to create a unified SARA that takes all these criticisms into consideration, and this might be a very useful task for the SCA Global and ARCA Global community. In my final slide, I just give a list of all members of the Clinical Outcome Assessment Working Group, and with this, I would like to thank you for your attention.
Okay, thank you very much, Thomas. That's a really thorough and very thoughtful overview in the context of SCA Global. So there are two questions. There is one saying, where do we find the online SARA training tool? Thomas, could you answer that?
We will send a mail immediately after the meeting to all members giving detailed instructions.
Very good. So please check the mail.
Yeah, so if you're registered as a SCA Global or ARCA Global, and we will extend that to the members of the European Reference Networks for Rare Diseases, we will use this email list. We already sent emails around, but we will give very precise informations where to find the training tool.
Very good. So that was a question by Dr. Yun Peng. And the second question is from Henk Engel, Dr. Engel. So he said, why not adding an NFL biomarker in blood to the minimum data set? And so Mattis wants to respond.
I just want to say that this is going to be this is not a clinical outcome. So Thomas Klockgether was focusing just on the clinical outcome measures. This refers to the biomarker, molecular biomarker outcome measure, which is going to be the next working group session. And this having said, NFL measures is still costly. We can only, we are going to focus about blood samples, which analyze we're going to measure is going to be discussed then in that session, which is going to be the next one.
Thomas, do you have an additional comment?
Yeah, I think Mattis is completely right. And but with the harmonization that we are also intending for the biochemical markers, the first step is to harmonize and standardize the biosampling. And when you have the standardized materials, you can do then all the lab studies which you're intending. But the important step for the harmonization and standardization is the standardization of the sampling procedures.
Okay, we have one more minute. Does anybody else have burning question? That's
maybe I can put one question because I'm not allowed
sure.
as a panelist to type in Q&A. Thomas,
okay.
Do you think we should make it mandatory for all SCA and ARCA Global participants to have a certificate in the SCA Global in the SARA training tool? Should we make it mandatory that we all complete it once just to reduce inter-rater variability?
Yes, I would suggest that if we have when we're starting SCA Global or ARCA Global endorsed natural history study, we should ask for that. We have requests from a number of industry companies, and they are at least interested in the tool. And the regulators, if SARA is used, they ask for that, and it's based on a large collection of videos. And I can add, even if we change the SARA, but this is something that will last some time, at least some years. We can still use this video material and for a new unified version of SARA. So it's a very useful tool, and it does not take too much time, and the certificate is not too difficult. So why not request that?
Very good. Well, time's up, so we're going to move on. Thank you very much, Thomas. And the next speaker does not need introduction, just like Dr. Klockgether. Next one is Dr. Gülin Öz from University of Minnesota about MRO biomarkers, and she chaired a session on that issue already.
So thank you very much again for this opportunity to tell you guys what we have been doing in the MR biomarkers working group. So just to remind everyone of our goals, first of all, our ultimate goal is to harmonize magnetic resonance imaging and spectroscopy data acquisition protocols for spinocerebellar ataxias across the globe. And to be able to do that, we are attempting to provide guidelines for a core as well as an advanced MRI-MRS protocol designed to obtain structural, biochemical, and functional biomarkers. So our guiding principles for these recommendations were two. The first one was inclusivity while maintaining a minimum standard of data quality. So we will not be taking essentially all comers of data, but we are defining minimum standards of data quality, but we also would like to be inclusive, for example, not only restricting the platforms to high fields, and the data quality standards, of course, may include some restrictions on hardware. The second principle is to make use of existing optimized protocols from other multi-site studies, not to try to reinvent the wheel, because there's a lot of work on that out there already. Similar to the clinical outcome assessment measures that Thomas presented, we are working with a core protocol and an advanced protocol, a minimum standard core protocol that would be able to obtain during a clinical imaging scan, and then an advanced, more research type protocol that not everyone may have the capability to administer, or even the infrastructure and funding to do. So our preferred platform for both of these protocols will be 3 Tesla. And when I go through the sites that are currently interested in this initiative, the reasons for that will be clear. But we also would like to establish a 1.5 Tesla protocol for sites who only have access to this platform. So what have you been doing since the face-to-face meeting, our last face-to-face meeting in Vegas? We've been holding bi-monthly teleconferences. We started out in June 2019, went through April 2020, and then we had a break during the summer, and then just had one couple of weeks ago in October of this year. In these teleconferences, we reviewed existing standardized MR protocols. We went over the ADNI studies, ADNI 2 and 3, the UK Biobank, the NIMS, this is the MS, North American Imaging Consortium in MS, and NICELS, that's the ALS consortium. We also had expert presentations. I listed some names here that spoke to our group and got their feedback, received their feedback and input in planning these protocols. We collected infrastructure information from the sites that would be immediately interested in participating in a global MRI-MRS effort. And so far we have information on 11 sites. They are listed over here. And all of these sites have 3 Tesla scanners that are accessible to any ataxia work. And all but 2 have Siemens scanners. So this is important to keep in mind. So while we will be providing general guidelines for a 1.5 Tesla platform, at least the sites that are so far interested in contributing all seem to have access to 3 Tesla scanners. And we have participation in the group from 30 institutions so far and 10 companies in 13 countries. So through our discussions, it quickly became clear that we needed some subspecialty working groups, subcommittees that would focus on these different advanced MRI, MRS modalities, because to provide detailed guidelines really needs that expertise on that specific modality. And we decided to include these 4 MR modalities in the advanced protocol. They include diffusion, magnetic resonance spectroscopy, resting state fMRI, and quantitative susceptibility mapping. And I'm listing the leads of these subcommittees who have been working on protocol guidelines for these modalities. And to do this work, we have been using the open science framework for exchange between the subcommittee members, as well as exchange with the larger working group. Open Science Framework has a lot of advantages. It's a secure cloud-based management platform. It has structured projects. It keeps track of versions of files that are uploaded. And the really nice thing about it is right now we have a private project, but at some point we plan to choose to open a subsection of that project to actually have appendices that can be updated over time, and that will be cited from the consensus manuscript that we will be preparing as a result of this working group effort. So this is how it looks like. This is just our SCA Global MR page. All the contributors are here. So you can not only exchange the MR protocols and of course also some presentations, relevant papers, etc. And we have the subgroup links here, and in each case, the participants can have discussions. This is what this bubble indicates, discussions about the protocols that are uploaded there by the leads of each group. So we discussed how to go about the consensus paper. We plan to have a core author group which will be composed of all the subcommittee members, and we will have a collaborator group from all the participants in the MR biomarker group. And the collaborator group, in fact, is listed in PubMed sub-citation, so everybody gets credit. We discussed several journal options. I believe in the end we will go with cerebellum, but this is still up for discussion, for a series publication of all SCA Global Working Group papers. This is one of the decisions we need to make with the larger group. In terms of content of the consensus paper, first of all, of course, we will have recommended protocols. And for the protocols, the general guidelines and important parameters will be listed in the paper. But then we would like to include very detailed, full example parameter sets that are platform specific in open science frameworks, so that a newcomer can essentially take that protocol if they have a similar platform and then have a good starting point for their research or their basic core protocol. We may include some analysis results because resolution, especially for structural images, is an open question. Different groups use different resolutions, and in the end, we probably will need to combine these, but we started to do some analyses from data that are acquired in the same brain from brains from the different multi-site studies, for example, ReadySky and ESME, and see if that analysis can provide some insights. And then we hope to provide some example data sets that are acquired on the main clinical platforms with the recommended parameter sets. So for the general MR protocol guidelines, we quickly realized that it is not possible to fully replicate fixed parameters across all vendor platforms. So we need to allow ranges in these recommendations. And what I mean by that, so this is a very basic table that goes over the basic and advanced protocol. As I said, for field strength, we prefer the 3 Tesla, but we would be accepting 1.5 Tesla data for only the basic protocol. As standard for clinical scanners, we will go with body coil transmit, but require multi-channel receive arrays. And there, this is a range that we had to define for the hardware component, coils with anywhere from 12 to 64 channels, because not everyone has access to a specific, for example, 32-channel coil. Coverage would be whole brain. For basic protocol, we decided to only stick to the structural T1 and T2 images with 1 millimeter isotropic resolution at 1.5 Tesla and 0.8 to 1 millimeter isotropic resolution at 3 Tesla. We had long discussions about potentially including a diffusion acquisition, quick, short diffusion acquisition, but the point was raised that even that may be challenging for sites that would be acquiring these data as part of a clinical imaging session, and they may not be funded. So this would be excluding some sites unnecessarily for the basic protocol. So we decided to only keep T1 and T2. For advanced protocol, as I said, we have, in addition to T1 and T2, we have diffusion, MRS, quantitative susceptibility mapping, and resting state fMRI. And I should say that we do not expect a site that is interested in running the advanced protocol to run all these 4 advanced modalities. A site may only be interested in doing diffusion or QSM or MRS, but these will be very valuable contributions for these modalities to a global effort. In the last few slides, I just have our draft tables for the main consensus paper, the protocol guidelines. I'm not going to go through them in detail, but just to highlight some aspects. For example, diffusion, we are recommending a 2x2x2 or better resolution. We have a range for echo time, a minimum acquisition, and multiband is an important consideration here with a range again. So for spectroscopy between single voxel and MRSI, we propose single voxel is preferred due to higher achievable data quality. And we have the recommendations for volumes of interest for single voxel. We have some recommendations on how to select the volumes of interest reproducibly, localization sequence, adjustments, water reference acquisition, etc. So the MRS component of these recommendations has been, I think, probably the easiest one, because over the last 5+ years, the spectroscopy field has been undergoing a lot of consensus efforts. I originally led this, and now different individuals are leading different technical pieces that contribute to spectroscopy. So we already have a couple of papers published, endorsed by this clinical spectroscopy community, and these are essentially replicating those recommendations. And then we have resting state fMRI. Again, for resolution, we have 3x3x3 or better resolution. Again, some ranges for TRT, and again, multiband. And finally, we have some recommendations for quantitative susceptibility mapping. You can read through these if you like. I'm not an expert in QSM, but luckily we should have 2 expert panelists who who are going to join me here. So if there are any specific questions, they can be addressed during the panel discussion. And then finally, I'd like to acknowledge the very active members of our group. I'm listing the 4 groups here with their subcommittee members. We have, I think on the call here on the teleconference, we have Dagmar, we have Andreas. Unfortunately, Ian couldn't make it due to the time difference. It's god awful time in Australia right now. Kune is here as a representative of industry perspective. He's from Pfizer, has been very active in this committee. And Jennifer, last but not least, Jennifer Faber has been co-leading this effort with me, and she's here with us as well. So looking forward to your questions and the panel session now. I'll stop.
Thank you. Thank you, Gülin. So it's open to discussion. I have not received any new questions with regards to the MR. So it's a panelist, please speak up and discuss further.
Anything I missed that you guys felt, oh, we should have mentioned this, please go ahead. The floor is yours.
If nobody's speaking up, I have a question. So there are so many MR part, particularly MRI part, there are so many variations. You know, it's kind of confusing. Is there any systematic nomenclature you're going to develop?
Systematic nomenclature for
Over what, how many channels and what the 3T or, you know, 1.5T or something like that. Based on those variables, can you just like at one glance, you know exactly what kind of protocol it was used?
That is very, I mean, that is difficult. We cannot really demand everyone to use 3 Tesla. We can have a very clear preference and encourage people, if they have access to 3 Tesla, for example, to go ahead and use
so
it.
that's exactly my point. So that you can make 2 versions, for example, ataxia MR, let's say 3T and 12 channel or something like that. That goes with a very systematic parameters and in the nomenclature, so that you can at one glance what kind of protocol it was used and what is comparable or not.
So I think maybe you're thinking about the protocol examples that we are intending to place in the Open Science Framework page. So those will be very specific. This protocol, for example, the ReadySCA protocol, we probably will share that. That's going to be for Siemens, Prisma, and Skyra scanners. So people will have access to it as a good starting point for at least some of these modalities, everything except QSM. So anything that we share online will be highly specific and tested on certain hardware, 3 Tesla with 32 channel receive coils, etc. The other advantage of that is, of course, you can update over time, because this is a moving target. New methods get developed all the time. So while frequent updates of these recommendations are, of course, not desirable, if, let's say, 5 years from now, we find that some of our recommendations are really outdated, we should be open to updating those recommendations.
Okay.
If others have thoughts, please go ahead.
A couple of new questions came in. One is from Dr. Anwi. Are there existing data supporting the inclusion of RS, resting state functional MRI, in ataxia trials?
There's very minimal data on resting state fMRI. That was one reason why we kind of deprioritized it in the ReadySCA protocol. We acquired as the last modality, and in some cases, people on certain medications, for example, we are unable to acquire it. But especially with several multi-site studies collecting it now, I think there will be more data. Yeah, the one big motivation, of course, is to collect structural and functional connectivity data together. When we were designing the ReadySCA study, for example, there was great encouragement from the National Institutes of Health to use protocols that were devised for the Human Connectome project. And of course, Human Connectome project, in addition to structural imaging, focuses on diffusion in resting state MRI. So there are all these data collected and databases built for control brains too. So it's an opportunity to use those 10 minutes at the end of a scanning session and get functional connectivity data.
Yeah, there's another question from Matis, Dr. Sinovsek. Sorry, Gülin, a bit off topic. Okay. Would the Open Science Framework platform a good opportunity for data and protocol sharing, also for the other SCA and the ARCA Global working groups?
I would say for protocol sharing, absolutely. Data sharing, now, when it comes to imaging data, that has very high demands for storage capacity. I highly doubt that OSF would be a good platform for MRI data sharing.
Okay.
And Gülin, say something, add something. I think, Gülin, very great work that you did there with your group, and you mentioned that you're planning publication, which I fully support. But we should have a common policy how to deal with these protocols. And I think one thing, they need to be openly available. And this can be at different sources, but we need to have a link on the SCA Global, ARCA Global website. As we should and will have it for the SARA training tool. But in addition, we should also publish the results of the working groups as papers. And there I would strongly be in favor that we show with the publications that these working group came from this global initiative and that they belong together. So they should be a joint publication or a series of publications in one journal. And we have also decided with the steering committee whether we still wait a bit and wait for the ARCA Global working groups, because I believe for the clinical outcomes, for the MRI and for the biosampling, the results will be very similar. So we will have results in a few months, and a consensus for all the additional things, digital assessment and what is just starting, it will still take some time. But then one could do a series of paper in one journal or one large publication with the consensus of ARCA and SCA Global.
Actually, this is Koen van Dijk speaking. I can take the next question that I see in the chat box. Bart van den Warrenburg is writing, I might have missed it, but will the Enigma initiative be used as an additional or related platform, or is it separate? So that's a great question. And unfortunately, Ian Harding is not on the call, but I can answer for him. Enigma ataxia is totally separate because it is retrospective analysis of data that is available, that people have already collected and are willing and able to contribute to a shared pool of data. And this SCA Global MRI protocol will be prospective. We hope that any group that starts looks at our recommendations and hopefully picks one of the protocols that are very, very specific. And if they have anything deviating, like they don't have a 32-channel coil, they might take a 12-channel coil, but get as close as possible. And then all the data will be collected and put together, giving in the end, hopefully, an even better data set than the one that Ian Harding is now analyzing. And I can throw the link to the ataxia, so Enigma ataxia group in the chat box, because that's a great other source. I hope that answered the question.
And just to add to that, while they are completely separate efforts, the Enigma experience will highly, highly benefit the SCA Global, this prospective initiative, because we will still be in a situation where we will have to combine data that were not acquired identically, like in ESME, like in ReadySCA, where you have identical protocols being acquired. And we cannot that restrictive, because then we would be excluding some currently existing active studies. For example, if we picked one resolution, that really would not work. So therefore, I think the Enigma experience will be very beneficial for us. And I just realized I might not have mentioned Dr. Christoph Langlet, who really has been very active in this effort too. He couldn't be here due to a registration glitch, and he's leading 2 of the subcommittees on diffusion and resting state.
Okay, very good. Any other comments, questions? Okay, so we have a couple minutes early. Then we can just move on. And if we finish early, there was some burning question for Dr. Klockgether, so we may come back if we have time. All right, so next speaker is Dr. Puneet Opal, and who is a professor at the Northwestern University in Chicago. And he has been a leader of a working group of biosampling standards.
I'm going to talk about the fluid biomarkers and the biosampling standards that we've been discussing. And coming at the end, I guess a lot of things have been sort of alluded to, but for the sake of continuity, I'm just going to go through how I thought about presenting the biosampling standards committee group findings. Okay, so as you know, we have the SCA Global right now, but in the last few weeks, we've been really working closely aligned with Mattis and Sinovsik and his group with ARCA Global. And you've seen the slide before, because actually it came across while we were talking about that it'll be great to align forces, because many of the things which we are going, whether it's for rating scales or biomarkers or imaging, are going to be in common, but especially for some of the fluid biomarkers, which I'm going to be talking about today. And we should really take advantage that the core operating procedures should be really in common. And while the biomarker candidates might vary, with the SCA being many of them, say, for example, targets for the polyglutamine diseases, and ARCA Global could be for the recessive, where you have to really raise the function of proteins. There will be something which will be common, for example, neurofilaments, which Mattis spoke about. And then also many of the scientists and clinicians are going to be working across platforms, so it gives other opportunities for us to interact. And I actually wanted to put the way we think about it moving forward early on, that we're going to have core global groups in SCA Global and ARCA Global. And in the SCA Global, I've tried to have an international representation. And we also, in the US, we have a natural history study, which is funded by the National Ataxia Foundation. And we are coordinating very closely with them, with the leadership of that. So Shenghan Core and Liana Rosenthal are on the National Ataxia Foundation core group. And then Alexandra Durr and Luis Pereira de Almeida and Julia Corelli are all going to be part of this, and they are available for the panel discussions later on. And for the ARCA Global, there's Matif, and he's recruited Filippo Santorelli and David Bengal as part of the ARCA core groups. And we hope that these core groups will at least set the major uniform standards which will align as we move into separate target and metabolite derangements later on. Okay, so biomarkers, as you know, are a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to exposure or intervention. With liquid biomarkers, everything is the same, except that now we're going to be looking at the liquid characteristics, whether it's blood, CSF,
Puneet, you're asked to use the full screen if possible.
You see it.
Doesn't matter, it's also fine this way.
Sorry about that, yeah. All right, so the goals of biomarkers are essentially twofold and maybe threefold. First would be target engagement for clinical trials. The second is surrogate markers for disease onset, severity, and progression. And then the third would be maybe genetic modifiers or novel pathways which you would get from discovery science. And now with these goals in mind, how do you sort of work backwards to get alignment of sampling and storage? So that's one point I wanted to raise about the goals, really. And these goals really take sort of immediacy when we figure out that we are in clinical trial readiness mode, where many of the SCAs in particular tend to be polyglutamine diseases, and they tend to be gain of function diseases where the target, the toxic protein, has to be potentially reduced. And there are companies which have identified strategies by which these can be reduced. And in all the major ones, we already have preclinical data in mouse studies where we know that we can really get these proteins reduced. So the question is, can we move on to a clinical trial, much like the Huntington field has moved on with Huntington disease, with ASO? So I wanted to just show this figure here, where this is an example of CSF and Huntington taken from CSF. And you can see a very nice trend of the Huntington protein coming down as a function of the dose of the ASOs. But I also want to point out in this slide that there's a lot of variability. These are just standard deviations, and they're huge error bars. Sample sizes are smaller, but frankly, in the SCA field, we will have even more of a challenge with sample size. So this is one of the things which is really going to come up when we are going to talk about ASO trials coming up in the near future. This is I want to remind you of what Matt has really presented earlier of his own work. But of course, there's neurofilaments which seem to be a strong predictor of degeneration, maybe just leaking out from damaged neurons, going relatively well aligned with SARA score, in fact, even doing better than it in many cases, relatively well aligned with age and also CEG repeat. But even between the cohorts, there's some variability. Some of the variability could come from, I'm sorry, the variability of different assays which are used, whether they are homebrew assays or others, but they could also be the fact that if the samples are not really well aligned in terms of how you store the samples or how you obtain the samples, this could create a challenge. One way to avoid the challenge is probably like what Martis did, where you can use one sample as a main cohort and one as a validation cohort, but it'd be nice if they can be kept as closely aligned as possible. And here is again some data from Mattis on the left and from our own lab on the right. And what Mattis showed on the left is basically mouse work where the variability is significantly reduced, obviously because now this is really what an ideal situation would look like, because in mice you basically have a single site really doing the work. And you can see there's very minimal variability, but also there's lack of biological variability as well. That's the ideal situation. But even in mice, on the right-hand side is our own SCA1 work. This is from SC1 mouse knock-in model. This model is a more severe model than the model of Mattis on the left, in the sense that these mice actually don't live beyond the age of 1 year. And you can actually see a very nice correlation. We don't actually reach the sort of ceiling effects or maybe the end results which you see with Mattis, but you actually can see that only later on. But we can see a very nice trend of increasing dose of neurofilament light chain in this SCA1 model. So this also answers one of the questions which was asked earlier about whether neurofilaments could be across the board, and I would say quite well, yes. Okay, so with humans, I want to remind you that we have many cohorts now. And the advantage of many cohorts is that we already know that we can work in multi-cohort manners. Each of these cohorts actually is a further branching out of multi-center sites. For example, the National Ataxia Foundation is a North American foundation with multiple sites, around 14, which are collecting natural history study, but also collecting right now biofluid markers. And ReadySky is also run by T. Ashizawa and Hank Paulson with SCA1 and SCA3 with international collaboration as well. The lower sites are basically the European sites. So there's Euroscar, RISCA, which you heard about, and ESMI. So with the advantage of these cohorts, what we did was we looked at all the protocols which have been, they're all available on their websites. And we basically made a matrix. And the matrix is what has been collected and in which tubes basically they've been collected. And in a way, the field of liquid markers is behind the field of our clinical outcome measures, in the sense that we are still at the point of basically keeping the samples, and we're making baby steps now, or maybe quicker steps now with Mattis's data, of trying to move this towards what metabolites we will analyze. But at least we know that these would be the standard standard sample which you would want every group to have when we go global. And that would be, we would want to have serum, because some proteins are better assayed in serum than plasma, which is the next on the list. We would want to have DNA, not only to confirm the CAG expansion, but if you want to look for modifiers down the road, for example, Huntington's disease, there are genetic modifiers which affect the CAG repeat length, for example. And then the rest below are somewhat optional. As you can see, there are some blanks in between, because we know they're difficult to do. For example, CSF is difficult because it's more invasive, even though it's a more proximate measure of what's going on in the fluid bathing the brain. It's a bit more difficult to get patients to volunteer spinal fluid, and it's also more expensive. The other ones are not so much as invasive, but they're expensive. For example, Buffycoat as a way to get RNA, Paxgene as a way to get RNA, and urine as a way, maybe it's easy to get, but we're not quite sure what we will do with it. So storage becomes an issue. And the same is also true with saliva or other bodily fluids, which you can keep, but we don't quite know whether we will use them down the road. So storage becomes an issue. So with this as a backdrop, we made a matrix of what we think would be a good SCA ARCA sampling protocol matrix, with the minimal being serum, plasma, and DNA, and an optional being CSF and PBMCs and RNA, dedicated RNA tubes. But what we would like, though, is that even with this minimum, we would not want to, one thing we don't want to compromise is the volume, because we would like this volume to be a resource for multiple investigations down the road. And we would suggest that we have at least 2 times 8 ml, so 16 ml of each of these samples. And the other thing which we don't want to compromise is the volume or aliquots, because we know now that many of these assays that we are doing can essentially be done on small volume with assays like Simoa. So can we store them at 0.5 ml aliquots, which would reduce the freeze thawing and therefore the variability which often comes from metabolites when the sample is exposed to freeze thaws, for example. For CSF, which I said was optional, we will still want to have, if we do CSF, we will want to have it at 0.5 ml sample aliquots stored in the freezer. We would also want to have follow-ups regularly, so this would have to be done, so you could do longitudinal studies down the road. So this would be our minimal, this would be the optional. We did look at many of the protocols which had different, I would say, challenges or criteria where they wanted patients to be fasting or a certain time of the day. We have sort of decided to keep this a little bit more flexible, because it's sometimes may be difficult for patients to come just dedicated for a certain sampling, and they might come for a doctor's visit later in the afternoon, for example, or sometimes early. So it may be difficult to get them in terms of specific time of day or in a relationship to a specific meal. And the equipment actually is also relatively minimal. Here we're thinking about, obviously, in North America and Europe, these would be, basic equipment. But let's say we want to go global in developing countries, all we really would need would be a freezer for -80 degrees and a clinical centrifuge, which is not would be a high-speed centrifuge. There was some comparisons across the protocols about how much you centrifuge these tubes, but we actually looked and we felt that as long as a center used one of the published protocols, which would be on either SME or Biosand, and Biosand, for those who are not familiar, is a US-based, basically a repository run by the NIH, which stores samples across neurological diseases, and it's funded by the NINDS. So they also have these protocols on their website. So a center which is not already sending it for sampling can very quickly be aligned with either a European protocol or a US protocol very quickly. So the good thing is that our protocols are relatively easily available and do not have to be reinvented. So moving ahead, I mentioned the fact that we are going to work closely with ARCA Global and SCA Global, and really makes sense to have all these intact, because let's say neurofilaments, it might be interesting to find out whether neurofilaments in the SCAs correlate with the neurofilaments in the recessive ataxias. Clearly the targets might be different, but there will be definitely points of overlap and comparison, which will be important not only for clinical trials, but also for basic science understanding down the road about cerebellar neurodegeneration in general. Other things which Gülin and also Tom mentioned earlier, consensus papers and website updates. It would be great to hook up to the SCA Global and keep these protocols and links and requests for materials and collaborations all in a singular website, with the real goal to be really mimicking or mirroring some of the other large-scale initiatives, whether it's ADNI, you probably have heard of some of these acronyms, but ADNI for Alzheimer's disease, Parkinson's for Parkinson's, and they have multiple. There's a PPMI, and run by Michael J. Fox, and a Parkinson's disease biomarker program in the US, run by the NIH, and I'm sure there are others in Europe. And Enroll HD, which is a polyglutamine disease, and I think is a forerunner to many of our ASO trials. So it will also be important not only to link it in terms of structurally with these initiatives from a operational point of view, but I also think that we might be able to do cross comparisons about the way things like neurofilaments, whether they're NFL light chains or metabolites, how they differ between different neurodegenerative disease, and tell us more about the disease, not just about clinical trial relevance, but also about basic science moving forward. So that's really where I wanted to go with the talk here, and I want just to say that I'm very optimistic about how we will move forward, and I think that this should be less tricky optimizing compared to what Gülin mentioned for the MRI, because we really have to focus on simple basic measures. And if we want to add additional measures, I leave it up to the leadership about whether we want to add urine, saliva, things like that down the road. And I'm happy to take any questions. And I have given the names of our panelists, and they can join for any panel discussion.
Okay, thank you, Puneet. There are a couple of questions. First is a link to clinical data, as well as probably the MR data. Second is, what about the distribution of the samples and storage of the samples? Do you have any comments on those? So those two questions, if you can answer.
So right now we do not have any links to the protocols, except that we can forward you to the links of Biosand, or we can forward you to the links of SME, but we don't have it all on the SCA Global platform, which I think would be the place to really have the link to protocols. In terms of sharing samples, we've had some discussions, and I know that they're sometimes difficult to get samples from Europe to the USA. And also informally, I've spoken to, just in my own research, trying to collate samples of different SCAs. I think most investigators I guess scientifically, they don't want to be just repositories. They really want to be involved in even the science. So I've heard from most people that they would want to be involved collaboratively. So this would make it probably more easier to do validation cohorts, and maybe like US could be a validation cohort for a European site or other way around. But I'm sure this will be more negotiated more when we have a specific question down the road, which will probably go through some sort of a vetting process about whether samples are discussed. And I know, for example, with NAF, actually, you know, that there's going to be a vetting process for Biosend, and I think it will probably have to go through individual vetting processes, and will have to be aligned with the SCA Global. So that is something which we have to probably discuss as a group.
Theo muted. Theo's still.
I'm sorry. So any thoughts about a link to the clinical data and MR data?
Yes, we have to move towards that. It's just that we haven't.
Yeah, okay.
Yeah.
So that's no brainer. Okay, so then I have a question, so that when you distribute, the great thing is that this is a great for the biomarker and other studies, but the problem is that samples are finite. And this is probably the last opportunity to do study sort of natural history data in both recessive and also dominant ataxia, because clinical trials opportunities will be expanded. So once they participate in clinical trials, no longer that's not really the natural history samples. So how do you, of course, new patients will come up, you know, with the new onset to the patients, but it is important to have clinical data linked and MR data linked, sufficient amount of biomarker samples. What do you think about that volume that you're proposing? Is that sufficient?
Well, you hit on all the points, T, basically, that we are going to have a lot of potentially even friction down the road, especially when the samples become smaller, and even early on about who gets the samples to do what particular biomarkers. So I think we will probably have to have a very strong-armed committee which will have to vet the studies. And frankly, I think that this will require not just a biomarker committee, it might even require Tom and Magis or someone like that to also throw weight behind it. This will also be a big issue when it comes to industries, when they want samples, that we have not even discussed that issue. The other question about volume, obviously the more volume you have, the better it is, but we have to be practical. And so we actually, the amount we are suggesting now is more than the amount that Biosend typically would have. So I'm willing to even negotiate more, but it's going to come down to how much will a patient give, and also how much we can store reasonably in small aliquots. So I guess these would be, yeah, I agree, they are sort of derivative secondary questions, and frankly, we need to have basically more discussions, and I think that this only be accelerated now that we're getting more and more closer to clinical trial readiness, but you have hit the right points. But so we will be having more of these discussions, and we'll probably have to, I think the biomarker committee will probably have to have even a more centralized relationship with maybe Tom or you about how we will negotiate who gets what samples and how much to parse out.
Yeah, I think this is one of the first topics we have to focus on in our next upcoming biomarker conference, web conference, and put this very high on the agenda. Yeah. And the amount of the volumes amount which we chose were really like the threshold, more would have received too much criticism from all the centers and from the ethics protocols. So I really think we were on the edge on how much we can sample. from our previous consortia in Europe, if you go higher, you have the experience that ethics committees will ask many, many questions. And so I think that is the upper edge of how far you can go without having all the centers asking questions. So I think that is that threshold.
I also think that once we discuss this, we'll have to go back to our individual repositories. So for in the US, we'll have to go back to Biosense. So there are some legacy issues. I don't think that the US is going to change where we send our samples just because it takes a lot of effort to basically negotiate the repository. I'm sure the same thing is true in Europe. And each of them, and also with Biosense, we probably have to also be compliant with how the NIH wants us to share our resources. So we might have to negotiate with yet another partner. So it may be more complicated than just even an internal SCA global negotiation. But I think but you're right, we have to keep them on the top of our list as so these can be addressed earlier rather than later.
Okay, any other comments? So I have one more question. So, obviously, biomarkers, target engagement is very important, and the disease activity, pathogenic mechanism activities, as well as surrogate to the clinical manifestations, those things are very important. But everybody is thinking in the same fashion, and you can receive 3 or 4 different groups, academics, pharmaceutical, and exactly the same kind of biomarker assays, and they want to have that biosamples for. And do you have any thoughts about sort of streamline those things with a consolidated effort? I know there's a CRC, SCA, NAF group is working with the industry to putting those people together to work together, but I don't know the details of those things. And what do you have any comments?
So I can give my thoughts, and then other people can chip in. I think for some of the standard ones like the targets, and now maybe the neurofilaments, if more people are interested, I would say that we should just team up. We should do it as a group, and we can put multiple people on the paper, and maybe we might have to have a leadership committee. But I think that, to me, the most sensible thing would be to team up rather than duplicating for a small precious sample. But I'm willing to listen to what other people have to say.
Okay. Anybody else? Let me check the box one more time. There was earlier a question about fasting. Does it make any difference?
I guess theoretically it might make a difference because that's why people in the Alzheimer's field, for example, their levels of A beta and their protein can follow circadian rhythms. At least that's what I read in the literature. But in our case, we are still in the discovery phase, so we don't know if it follows a difference. But I think our hope would be to find robust biomarkers initially, and we don't want perfection to mar the fact of getting something good out of it. And if you don't have sufficient samples to begin with, because we have too many stringent criteria, you might not just get samples in sufficient quantity. So that's how our thinking went.
Okay, anybody else? If not, we're going to go to the next speaker, and SCA Global Policy by Holm Graessner. And he has been really great in administrative aspect of the SCA Global, and including the policy and regulations. Holm, are you on? Okay.
Yes, thank you, T. I'm going to report on behalf of the SCA Global Working Group Policy. And the tasks given to the Working Group Policy at the Las Vegas meeting were first to come up with a membership policy, so the general principles to abide by to be a member, to become a member. Then second, to give guidance on topics of interest, and we touched on the informed consent from standards and on publication policy. And in the time between Las Vegas and now, 2 new tasks came up. One is the collaboration with the critical path for ataxia therapies, about which we will hear tomorrow further things and what the status is. And as alluded to already quite a bit, communication was a task also, which is not really policy, but is important to have a reference point where all the standards, harmonization, protocols, etc. Can be linked to, and where the resources of SCA Global, but also of course of ARCA Global can be found. So we don't start from zero. We have a steering committee, which is composed of Tia Shizawa, Alexander Dürr, Laura Yadim, Thomas Klockgether, Bing Song, David Smuljewitz, Julie Greenfield, Sue Hagen, and me acting as the secretary. So let me start with the proposal for the membership policy, and be really focused on individual clinicians and researchers as well as patient organization, and that you will see, and that's a bit of a deviation what we initially had in mind when we thought that we could also focus on institutions, but this is really now for researchers and clinicians as well as patient organizations. And then the first thing and the first principles we SCA Global and all the people working at SCA Global should agree on and abide by, of course, the goals. And those goals Thomas mentioned, and you might recall that at the meeting in Las Vegas, and I just read the goals. To provide added value through a global SCA research platform that is based on exchange and sharing, to establish large, well-characterized, standardized trial-ready cohorts, to precisely define the evolution of clinical outcomes across all SCAs and different world regions, to better characterize the pre-ataxia stage of all SCAs, and to develop and validate imaging and biochemical biomarkers, and last but not least, to attract young talent and improve education and training of clinical investigators. And as Thomas already said, and Mattis mentioned that as well, so SCA and also ARCA Global is really about facilitating and moving things ahead, and not a funding organization. It's really a catalysator and doesn't provide any funding. So these goals mentioned here are really focused on harmonization, facilitation, and providing guidance. So what are the principles of collaboration? And that's the second bit we discussed. So first, SCA Global research should be collaborative. Members of SCA Global should aim for sharing resources, data, and material in the SCA Global Consortium. Enabling local frameworks should be aimed for. So that's one of the things we touched also already on, and I think it's not yet clarified to a point that we can really start sharing resources, data, and material. So we believe that there's still guidance to be given, and as you will see, we're going to propose to set up a new working group and on exactly this topic. The second principle for collaboration, SCA Global research should engage patients and or their representatives in all relevant aspects of the research. Third, international, national, regional, and local legislation regulations need to be adhered to. Fourth, SCA Global investigators agree to adhere to SCA Global standards and harmonization with respect to subject data, clinical outcome measures, imaging procedures, informed consent components, and biosampling. 5, SCA Global investigators agree to facilitate collaboration with third-party industry partners. And 6, SCA Global partners aim to publish all results in a timely manner in peer-reviewed scientific journals. So what about governance? And I mentioned already that we have a steering committee already, which have been put in place before the Las Vegas meeting. But we really discussed what the principle of governance could be. So on participation, investigators of ataxia clinical research sites and patient organizations can participate. And I'm going to discuss how that relates to the participation of industry participants. Then 3.2, investigators of ataxia clinical research sites will have to fulfill clinical scientific qualification criteria. So basically, we don't want anybody to become a member, but we will have qualification criteria which those people who want to join have to comply with. These criteria will be defined in detail by the steering committee of SCA Global, and as you will see, it's then SCA and ARCA Global together. And next, in order to participate, investigators will have to approve and sign this policy document. So it's really in individual abidance to these principles I just introducing. So those were the criteria for the researchers and clinicians. The second main group being members of the SCA Global initiatives are patient organizations, and for those we also discussed criteria. So first, being equal partners in SCA Global research. Second, agreeing to endorse SCA Global. Third, willingness to publicize the initiative amongst ataxia clinical experts to encourage engagement of sites. Fourth, dissemination of information to patient and patient organizations worldwide on SCA Global. Then to encourage cooperation among SCA patient organizations worldwide, being a link to existing patient groups globally, be prepared to be involved in providing the patient voice in clinical research and trials, willingness to consider support in organizing meetings and conferences, facilitating contact and discussions with potential funders where possible, so for instance, pharma companies, and represent the patient organization in the SCA Global Steering Committee. And these patients organizations, which are members of the initiative, will be listed, and they will be included in the email distribution lists and being kept informed about the developments. And as you see, quite a number of these principles we adhere to already, and this is in a sense the summary of things we have partially achieved already. So governance, and that's reflecting that SCA Global should really be a global initiative. So the steering committee of SCA Global will be composed of representatives from each continent. So for now, Europe, Asia, Africa, North America, Central and South America, Australia and Oceania. And the election procedure will not be done as a top-down procedure, but by the respective continental groups, which will determine who will represent the respective continental group in the steering committee. And in addition to these representatives, patient advocacy groups, so specifically 2 will also be members of the steering committee. What's the mandate of the steering committee? It's to establish stipulation and processes that determine the collaborative framework of SCA Global and to endorse SCA Global projects and resources, as well as represent SCA Global in the communication to and with external partners. The SCA steering committee is also mandated to establish working groups and to appoint leaders and co-leaders. And here, really the participation of industry persons, of industry representatives comes in, because working groups are open to participation of persons with express interest and are accepted by the respective leaders of the working groups. And as we have seen in the working groups, and I think most prominently in the MR imaging working group, this was also already kind of achieved and actually brought about very nice results. The steering committee will elect a chair and a co-chair, and the chair and the co-chair will coordinate the steering committee in fulfillment of their mandates. And the steering committee will appoint a secretary, which will coordinate the operative implementation of the decisions. All mandates will be for 2 years and will be renewable. So a few comments on the government structure. So we will make available a full list of members on the website as soon as the continental groups have, according to the membership criteria, put together those lists. Then, as said, the continental groups will self-organize themselves. So it will not a top-down, but rather a really bottom-up process. We might consider to further divide the continental group depending on the development of the field. As alluded to already, the working group leaders will set up rules to allow non-SCA global investigators to participate in the working groups. And this very specifically relates to industry investigators. On collaboration with external partners, so the SCA Global Consortium aims at collaborating with third parties, and the most striking example, of course, are the industry companies, so the pharma companies, but also biomarker companies, but also, and that I touched on already, the critical path initiatives. For SCA Global related collaboration with any particular third-party member should abide by a process being set up by the steering committee that will be based on the principles of transparency, review, and endorsement. And last but not least, and that's what I mentioned already, and what I think is yet unfinished business on data storage, sharing, and protection, as well as material exchange. And you all know that is a tricky field. So we will try to set up a coordinated approach to enable data sharing and the material exchange. As Puneet already mentioned, that's really also dependent on the local, national, and well, mainly national and also continental in terms of Europe stipulation setup. Then SCA Global investigators aim to set up a respective local frameworks to enable data sharing and material exchange. So it's really dependent on, firstly, on the investigators on site to enable data sharing and material exchange. Then the SCA Global projects and partners aim to adhere to standards endorsed by SCA Global. SCA Global investigators aim to store data according to SCA Global data standards locally and in SCA Global registries or registry. And of course, in order to do that, in order to be able to share data and biomaterial, the SCA Global investigators strive to implement the SCA Global informed consent standards. So we have a few points for consideration. The first point is the representation of basic research in the steering committee, which is not yet in place. Then the representation of working groups in the steering committee, including the Young Investigator Initiative. As I said, so we would propose to establish a working group for data and biomaterial sharing for facilitating this sharing and setting standards. Action points. So the first action point, and Mattis and Thomas and Puneet mentioned that, is form one steering committee of SCA Global and ARCA Global to align activities and governance. So from our perspective, it doesn't make any sense to have separate steering committees, but really one steering committee, one overarching steering committee should be formed and should kind of steer the entire global ataxia initiative. Then for the current steering committee, discuss, amend and improve the membership policy by end of this year. Then to establish the membership criteria so that we are able to really determine which researchers, clinicians and patient organizations are the respective members of the initiative. Form continental groups, and that's up to you guys, and elect representatives for the steering committee by mid of 2021. For the steering committee and the secretary is really then to establish a membership. So the list of members I mentioned already by mid of 2021. So on guidance, the second main topic. So the first guidance we discussed was the informed consent standards. And we discussed that a bit and discussed the specific elements for SCA Global that could be considered in local informed consent, consents being used. So the first specific element was the storage of data in SCA Global registries. So if I say specific elements that should be considered, it merely means that those elements should be used in consents, patient consents that are being used at the local sites or in studies which will be or are endorsed by SCA Global and ARCA Global. Second, the access to data and samples for SCA Global researchers purposes should be in the patient consent. So who will have access, who should control, and what the procedure in place is. Then the possible linkage to different SCA Global data. And that mainly refers to a unique identifier to be used so that we can link what is being done in the different studies and can link the data in the different registries. Fourth thing is secondary use of data and samples for additional research projects. Fifth, the possibility of data sharing across SCA Global research groups. And the sixth is sample and data access by industry. Regarding publication policy, it's mainly that the publications should adhere to good scientific practice and acknowledge SCA Global. So the action point here would be if agreed upon by the steering committee to establish a working group on data and biomaterial sharing to provide a platform service, so to really facilitate and provide standards on this topic. So the third main point is the collaboration with the Critical Path initiative. So as hopefully many of you know, we have established a collaboration with the Critical Path for Ataxia Therapies. That's in the process being formed now. The aim of CPAT is really to establish regulatory improved outcome parameters for clinical trials. And we thought about the role SCA ARCA Global could play in respect to CPAT. And we think that SCA ARCA Global working groups should really provide the academic consensus for the CPAT working groups. So in a way, feed the academic consensus as a content for the discussion in CPAT, and also in terms of representation of the academic field, determine the academic membership in CPAT. And then in terms of how this could work in terms of data, the idea would be that there's a coordination of data provision, production, and needs. So SCA Global partners provide data, retrospective data, so CPAT. CPAT utilizes the data and also determines potential data needs. And SCA Global is then to respond, if possible, to the respective data needs being expressed by CPAT. So last but not least, on communication, this is just a summary of what has been done, and I think which has referred to a bit already. So we have a website for the Ataxia Global Initiative, and you see the link there. But if you just type Ataxia Global Initiative in whatever search machine you use, you will find it. This Ataxia Global Initiative has two subsites. One is for SCA Global and one is for ARCA Global. And part of this of these sites is a space for resources, and in this space for resources, we can of course put all the protocols, all the links we have agreed on, and use it there as the main and the central point of reference for the Ataxia Global Initiative. Then we have, of course, a Twitter account, and you can tweet on Ataxia Global and also refer to Ataxia Global. And of course, the last bit for the communication topic was the organization of the ARCA Global Conference itself, which we are now in, and with that, I would like to finish.
Okay, well, thank you, Holm. We have several questions here, and from Dr. Georgian, and I don't know, the question disappeared, if I remember correctly. She asked, since investigators and SCA Global to do some research activities, they have to apply for the approval from a local ethics committee and government and other things. So would SCA Global establish sort of a consent form, ICF or protocol, and that will be available to the investigators?
So we discussed that, and all the other person on the panel now, please comment on what I say. So we discussed it in the working group, and the consensus was that we would like to give guidance on the first hand, on the one hand, on what topics to cover in the consent form, but also we wanted to come up with the exemplary consent form. And we haven't done that yet, but I think that's still on the table, and I think it might be very useful to have an exemplary consent form which can be used as orientation, and then be modified according to the local national stipulations.
Okay, another question is from Vikram Shakote. He said, I like the architecture of governance of SCA ARCA Global. Since this is not a funding organization, funding agencies will have to be brought in to help investigators adhere to the guidelines. If pharma provides funding, is there an administrative role that the SCA and the ARCA Global can take to facilitate conversations with funding agencies?
So yeah, good question, and maybe also a tricky one. So I could imagine that if a study or a project is being endorsed by SCA or ARCA Global, that might, or maybe even an application in a way that might serve as a positive point for the application. However, I think we will not to interfere with the evaluation of a funding agency. So I think funding by a funding agency and endorsement by the SCA Global or ARCA Global initiative are two different things. Yeah, Thomas.
Okay.
I have a number of comments or questions. First relates to participation. If I understand correctly, the plan is to have individual investigators, but on the other hand, patient organizations. So it's a different type of members. One are organizations and the other are individual persons.
Yeah, correct. That's yeah, that's how we discussed it, right?
Okay. And you feel that this is appropriate?
Yeah, perhaps. I mean, perhaps Julie and Sue can comment on that. So my thinking on that would be that the patient organization are being represented by members of those patient organizations, and then in a way we are on the same level again.
Okay, so
yeah, but on the other hand, this is then you can also say I'm representing University of Bonn or DZNE, but this was then finally the decision not to go this way because it would make things very complicated because you would then have to have contracts with all the organizations. So why don't we handle it in the same way with the patient organizations so that Julie is member and there could be also 2 or 3 other members coming from UK ataxia.
So
all members of UK ataxia will apply for membership in SCA Global.
I suppose the roles are quite different though, aren't they? So at the moment we're saying that the patient organisation roles are kind of disseminating and promoting and those kinds of roles. So whereas the investigators' roles are very, very different. But you might get many, many members if you allow all patients to be members. You might end up with a very large number of members, but that might not be a bad thing.
Yeah, but we could also define criteria. So we will not also include all neurologists worldwide. We have certain criteria, and so for representatives of patient organizations, we can also say we accept only those who have leading positions and expertise in interacting with academic researchers and so on. I find it a bit awkward to have one organization and we have type I members who are individuals and type B members who are organizations.
yeah, so I would yeah. So just listening to you, I would be inclined to really consider that and to discuss that in the working group again. And I agree. It's so have it on to have it on the same level. So have to have individual clinicians and researchers on the one hand, and to have also individual representatives of patient organization. I think would be consistent. But we haven't discussed that yet. So that would be an action point for the working group to define, at least as a proposal, those criteria, if the group can agree to this proposal.
Okay, another point also related to participation. We need to. You mentioned that we still need to define criteria. What is important that we have criteria that also allow young investigators to join and participate. And if you have a criterion needs to have 5 publications in the ataxia field and 5 years experience in ataxia clinic, we will not be able to attract the young investigators, and this is a major goal. So this needs to be considered.
So that's going to be a next session, SCA Global Young Investigator Initiative.
Yeah, I think we can rely on the on a good discussion and a good decision by the steering committee on defining the membership criteria and to introduce different levels of criteria for senior and young investigators.
Okay.
Yeah, I think this is also routine in other societies, movement disorder society. They also have these young members and senior members and these things, yes.
So there are a couple more comments and questions. One is Dr. Richard Roxball, and this is more similar to treat NMD. They are 10 years down the track in this sort of organization. We can learn lots from them too. I think also Huntington Study Group, they have sort of a similar route, and CRC-SCA NAF invited him to discuss. But do you have any idea, Holm?
So on I can see that know quite well the NMD leadership, and we are on different initiatives and projects in quite tight collaboration. So with people from Newcastle, basically, who lead on NMD, and we are quite aware of what they've done. They have a number of things which we can learn from. But as mentioned, they are 10 years ahead. So it's a bit of way for us to go still.
Okay. Another question from Dr. Schmitz-Hübsch. How do you envision the conditions of industry collaborations? How would you deal with collaborations of SCA Global members, for example, biomarker testing or else?
Yeah, that's why in the stipulation for the participation in the working groups, it was mentioned that the working group leads are really to define the respective criteria for membership in a transparent and open way. And I think this is important also because we want to avoid this conflict of interest. And I think it should be very clear on what basis industry participates in the working groups, and I think it should be also transparent which industry participates. And if you do that, I think we should be able to avoid conflict of interest. If not, I think it's for the leads of the respective working groups to deal with it. And then if they are not able to or provide or would like to have assistance, they can refer to the steering committee.